The overall goal of the following experiment is to conduct hybridization in situ for determining the localization and accumulation patterns of specific transcripts within mosquito tissues and embryos. This is achieved by first fixing the mosquito tissues or embryos so that the tissue ultrastructure and mRNA targets will be preserved and available for hybridizing with antisense RNA probes. Next, the antisense RNA probes are hybridized to the target transcripts within the fixed tissues or embryos. Then, colorimetric antibody staining methods are performed in order to detect target mRNA localization and accumulation patterns. Results are obtained that show spatial patterns of RNA accumulation based on hybridization in situ and colorimetric detection methods.
This method can help answer key questions in the field of functional genomics, such as addressing when and where gene products accumulate in the mosquito. This procedure also allows us to obtain spatial localization information to understand better how gene expression is regulated for biological processes such as reproduction, development, and mosquito pathogen interactions.
To prepare for hybridizing digoxygenin-labeled RNA probes to mosquito tissues, begin by dissecting the ovaries and salivary glands. Then, fix the tissue and treat it with proteinase K if necessary. After the tissues have been post-fixed, equilibrate them in 1 milliliter of a 1-to-1 mixture of hybridization solution PBT or Hyb PBT for 30 minutes with nutation.